welcome to my channel. Today's video is on respiratory complex 1 and associated mitochondrial diseases. Mitochondria is the powerhouse of the cell where most of the respiration occurs to generate ATP as a form of energy. ATP stands for adenosine triphosphate, which is then used to carry out different metabolic activities in the cell. So now let's have a look at what respiratory complex 1 really is. Respiratory complex 1 is the first enzyme complex in the electron transport chain. Electron transport chain is the third step in the respiration. Complex 1 is known as NADH ubiquinol reductase. The role of complex 1 is to reduce NADH by removing electrons from it. The NADH can be oxidized back out later in the respiration. The energy from this redox reaction is used to transport electrons across the inner membrane. The electrons from complex 1 are transported to complex 2 and subsequently to complex 4. At each complex, similar redox reaction occur, and the energy from those can be used to transport electrons across the membrane. The buildup of proton gradient across the membrane is the main driving force behind the production of ATP. So now let's have a look at what the structure of complex 1 is. The structure of complex 1 is, it is an L-shaped structure with two perpendicular arms, one protruding out of the matrix and is known as peripheral arm, composed of hydrophilic subunits. The other is known as membrane arm and is embedded in the inner membrane and is composed of hydrophobic subunits. The minimal working model is composed of 14 subunits and is called the core structure and is conserved through many species, out of which 7 are from peripheral arm, which are nuclear DNA encoded, and 7 are from membrane arm, which are mitochondrial DNA encoded. Some of the mitochondrial diseases that are linked with complex 1 are such as Lee syndrome and myoclonic epilepsy. Most of the tissues that are affected, or the muscles that are affected, are the ones that are high energy demanding, such as in Lee syndrome, which is a neurological disorder. It affects the mental and movement abilities, leading to an early death. Other diseases that have been recently linked with complex 1 deficiencies are such as diabetes and aging. One of the causes of mitochondrial disease is the dysfunction of complex 1, which can be due to mutations. The natural occurring mutations are called spontaneous mutations that can be due to a mutagen such as GV light, which affects normal gene and knocking out an amino acid or changing it for another one, resulting in a mutated gene. The mutated gene then transcribes into a malfunctioning protein, which is not able to or less able to function as a normal protein, resulting into the disease. However, to study the effect of certain mutations, site-directed mutagenesis is used to introduce mutations experimentally. In site-directed mutagenesis, a DNA template is taken, which is a double-stranded. You heat it to denature it and to get a single-stranded DNA template, which is called a primer. The DNA single-stranded plus mutagenic primer, which is mixed with it, will give you a mutated DNA, as it will anneal to the single-stranded DNA at a very specific sequence, which can then be extended using enzymes such as DNA polymerase. That will give us mutated DNA. In conclusion, there is a correlation between the diseases that have been mentioned and dysfunctioning or deficiency of complex 1. However, more research is required to see what functional